All right, we're going to talk about homeostasis. Homeostasis is basically an organism's ability to maintain a variable within a specific range. To do that, it has a set point. The set point is the ideal spot for a variable to work in. So we're going to call this our set point right here. It also has a normal range. And the normal range is an acceptable fluctuation that a variable can work in. So ideally we want to be on the set point all the time, but what ends up happening is we're going to be up and down within this normal range right around our set point. To do this, our body has what's called a homeostatic control system. Our homeostatic control system is made up of a few different things. We can just write them over here. So we have homeostatic, that might be a touch high. Homeostatic control. And what we have is a stimulus, a receptor, an afferent pathway, a control center, an efferent pathway, and effectors. And I'm going to try to explain how these all work together to help maintain homeostasis. So let's take, for example, body temperature. So we know that our body temperature, our set point is 98.6, but let's say we can go a degree lower, so we'll just say 97, and then up here we'll just say 100 degrees. We'll say those are in our normal range. So what happens, this is our set point. So what happens if it's cold outside? If it's cold outside, we're not wearing a jacket, the stimulus will be the temperature outside, so it's cold. So our body, we have receptors on our skin that say, hey, it's cold, our body temperature's starting to go down. And it's starting to go down, so this receptors, it takes that information along the afferent pathway to the control center. The control center here is our hypothalamus in our brain. And it tells our control center, hey, our body temperature is starting to go down. And so our control center, it makes a choice. It says, okay, we need to do something about this. So what it does is it sends a message along the efferent pathway to the effectors. Now in this case, effectors can be our muscles. It can also be peripheral resistance, so the diameter of our arteries in our extremities. We're just going to call that peripheral resistance, PR. And these are what are going to try to reverse this temperature decrease in our body. So our muscles are going to start shivering, trying to warm us up, and then the peripheral restriction, it's going to push more blood into our core so that we don't have quite as many things cooling off the body. We don't have the effects of evaporation. And what that's going to do is it's going to bring us slowly back up to our set point at 98 degrees. Contrarily, if we go too hot, if we are out in the middle of July, 100 degrees, and we are running a marathon, and we start to get too hot, our body's going to say, all right, it's too hot, we need to make some changes. So the stimulus is too hot, our receptors are going to say, hey, brain, it's too hot outside, we're getting a little warm, our body's getting a little warm. It's going to send a message to the efferent pathway to the effectors. Peripheral resistance would be an effector in this case, and also sweating. So our peripheral resistance is going to be, our arteries are going to be open, so they're going to be pushing blood out towards the extremities to try to take effect of evaporation and cooling, and also we're going to have some sweat to help in that as well, trying to once again bring us back towards our set point at 98.6 degrees. And I can show you on a graph just how that works. So we have our stimulus here. Our stimulus is going to act on a certain variable in our body. So we'll just say variable. And this variable is going outside of the normal range. Because of the stimulus, the receptors detect that. And they send a message along the efferent pathway to the control center. Which makes a decision to go out to the effectors. Which are then going to try to bring this variable back into the normal range. When it does this, or when it's trying to do this, it's called negative feedback. Most of the time, this is what the body is going to want to do. It's going to want to bring a variable back into the normal range. It's going to want to have negative feedback. There are a few cases in which positive feedback is beneficial. And what these are, instead of the effectors trying to maintain or bring back into the normal range, bring the variable back into the normal range, they are going to push the variable further and further outside of the normal range. And the body has a few of these that are beneficial. The one is childbirth. The next is blood clotting. And the third is childbirth, blood clotting, and action potentials. Childbirth, when a woman starts to go into labor, her cervix will start to stretch. That'll send a message back up to the brain. The brain will then make a decision. It'll release oxytocin, and oxytocin will actually promote contractions. More contractions bring more stretching at the cervix, and the cycle continues and continues until the child is delivered. So those are a couple examples of positive feedback with homeostasis. All right, the next thing we want to talk about is protein transport. In this, we have to understand that there is, this is a cell membrane, so we have the extracellular fluid here, we have the intracellular fluid here. And this membrane right here, it's made up of a phospholipid bilayer that is impermeable to anything that has a charge, anything that's polar, anything that's large, they can't get through. So what they need is a transport protein. And we'll just draw a transport protein here. And these transport proteins, they help these large polar molecules with charges, get to the intracellular fluid from the extracellular fluid. The first one that I want to talk about is passive transport. So passive transport, what it does is it follows the concentration gradient. So if, once again, if we have our cell over here, and if we have a large concentration of sodium out here and a small concentration of sodium in here, the concentration gradient would be from outside to inside. So sodium would want to go through here, but because it has a charge, because it's polar, it's not able to go in to the cell without use of a protein transport. So we're going to first talk about passive transport, and there's a couple different kinds of passive transport that goes through the cell membrane. The first one is channel. Channel proteins. And remember, all of these are going to go with their gradient. So they're either going to go from inside to outside or outside to inside. The second one is voltage gated. And the third one is mechanical gated. Because these want to go with their gradient, they need to have, most of these have a gate on them. So they'll have gates. So we'll draw, once again, our cell up here. So if we draw a channel protein, and it's going to have some sort of gate on it. And what happens is when something wants to come down here, so let's say we have a potassium. Actually, potassium is going to want to come out because there's a higher concentration of potassium on the inside of the cell. So potassium wants to come out. So what happens is potassium will come over here and it will bind. So these channels are specific to certain ions, certain molecules. They don't just let anything go through. A potassium channel is not going to let sodium go through it. The same with a calcium channel. It's not going to let sodium. It's not going to let potassium go through it. So this potassium molecule comes up here. It binds here, which changes the shape of the protein, allows the molecule to come out into the extracellular fluid. This is an example of a potassium leak channel. Another channel is a voltage gated channel. So once again, this is specific to a certain ion. When there is a change in the voltage of the intracellular fluid, it will cause a change in this protein. The gate will open. So let's see here, I'll just draw a gate like this. The gate will open, and it will allow whatever it is specific for, it will let it come out of the gate. The last one is mechanical gated, so anything, these are sensitive to touch, so like in our fingers, heat, anything like that will stimulate this gate to open, and once again it's specific for a certain ion, so like a calcium ion. Once it gets open, a calcium ion will want to go out or go in based on its concentration gradient. There's one more, also the ligand gated. Basically with the ligand gated, it requires something other than the molecule that's trying to go against its gradient. A common one we see is acetylcholine, so ACH, and it will bind to this ligand gated receptor. Once it's bound, the door will open, and the molecule will be able to move in or out based on its concentration gradient and the specificity of the channel. Okay, what else do we need to talk about? All right, we can talk about carrier proteins. This is another example of a passive transport. Once again, these are specific. Unlike the other channels that we've talked about, this is a carrier protein. And this one is not open all the way through at one time. So if we have two of our membrane here, we have our extracellular, our intracellular. If we have a molecule that the protein is specific for, what it will do, it will come in, it will bind here, the protein will change shape, and then it will open to the intracellular fluid. Once again, these are specific to certain molecules. A common one for this one is going to be GLUT receptors. So these are the GLUT receptors. These are what allow glucose into the cells of the body. The one thing about these receptors is that they can become saturated. So if we have a lot of glucose here, it needs to get in the cell. If we only have one or two receptors, one or two of these carrier proteins, they can become saturated. So they're working as hard as they can, but they're still not keeping up with the glucose molecules that are wanting to come through into the receptors, into the cell. You'll see this with diabetes. Patients will have an excess amount of glucose in the blood, and as it goes down into the kidney has certain carrier proteins that will basically take it and put it back into the blood. When these become saturated, you'll start to lose glucose in the urine. That's just an example of saturation. All right, what else? We need to talk about active transport. So active transport The difference is between passive transport and active transport. Active transport, you're taking molecules against their concentration gradient. So where we have something on the outside of the cell in a high concentration that wants to go inside, active transport is going to take what little we have on the inside and it's going to pump it outside, making this concentration gradient even greater. And that's important for setting up secondary active transport. But we'll talk about active transport first. So active transport, and we'll talk about an important one. So normally with our concentration gradient, we have a high concentration of sodium on the outside of the cell. So we'll have increased concentration of sodium, and we'll have a low concentration of potassium. So potassium is low in the extracellular fluid. We're just going to call this the extracellular fluid and the intracellular fluid. And on the intracellular fluid, we have a high concentration of potassium and a low concentration of sodium. So based on the concentration gradients, sodium is going to want to come into the cell to balance it out. Potassium is going to want to go out of the cell to balance it out. What we have with active transport, we're going to call this one our sodium potassium ATPase. So sodium potassium ATPase. So what happens is we can have ATP. So we have ATP come and it will bind onto this protein, and what it does is it will take 3 sodiums from inside the cell and 2 potassium from outside the cell, and it will use the ATP to pump the sodium out and the potassium in. And like I told you, this is important for setting up secondary active transport. So secondary active transport, what it does is it still requires energy, but it requires energy in the form of a concentration gradient from one to another. So if we just take this theoretically, we have a concentration gradient. Potassium wants to go out. So we could use potassium that wants to go out. We could use potassium that wants to go out through its concentration gradient, and we can use it. We can use the energy from it wanting to go out to bring something else either out with it. That's called symport. Or we can, let's draw another one here. Or we can have something against its concentration gradient. We can have it come to the intracellular fluid, and those are basically secondary active transport. All right. I did skip over diffusion. Diffusion is a passive, and I think I kind of mentioned it when I was discussing passive and active transport, but passive transport, we can talk about diffusion a little bit more. So diffusion is basically a molecule's ability to migrate across the cell. So like I said, these are going to be things that are nonpolar, things that are small, things that are fat soluble, so we're going to say like fatty acids, steroids, cholesterol. Steroids might not, they might be a little big, but cholesterol. These are things that can diffuse into the cell just based on the concentration gradient. With that, just diffusion in, there are a couple of factors that play a role into how quickly they can do it. This is called Fick's law. Okay, and Fick's law states, let me just see, refresh my mind. So J equals negative D A times concentration of C divided by the concentration of X, where J is the diffusion rate, D is the permeability constant, diffusion coefficient. D is the diffusion coefficient, A is the surface area of a membrane, C is the concentration difference, so basically from the inside to the outside, and then delta X is the thickness of the membrane. And we can use all that to determine how quickly the molecule will get diffused into the membrane. Okay, the last thing we're going to talk about is osmosis. Osmosis, tonicity. So osmosis is the movement of water across a semi-permeable membrane. And just a quick thing really quick, so we have molarity, and this is important in osmosis, so molarity versus osmolarity. It's important to remember, so molarity is the number of moles of something per liter of solution. Osmolarity is the number of particles of something per liter of solution. So that's important when you think about ions. If we added salt to a solution, NaCl, 1 mole of salt to a solution, we would have a molarity of one unit of salt. But an osmolarity, because it's in a solution, it's going to, the ionic bonds are going to break, and we're actually going to have a 2 molar osmolar solution, because the sodium and the chloride are each going to be their own thing. So, when we talk about osmosis and osmolarity, osmolarity is measuring how many particles of a molecule or particles of something are in a solution. So we can use osmolarity to compare 2 different solutions. So if we have 3 sodiums here and only 1 sodium over here, we can say that solution A is hyper osmotic to solution B. In the same way, we can say that solution B is hypo osmotic to solution A. If they had the same If they had the same number of particles in the solution, we would term them isoosmotic. Tonicity is a little bit different. Tonicity is comparing essentially our cells in our body to a solution that it is placed inside. Because the cell membrane is semi-permeable to water through channels called aquaporins, water will either exit the cell or enter the cell in order to balance the osmolarity. So if we have a solution, here is our big solution, and we place a cell in it, and the body cells are usually around 300 milliosmol. That's the measurement that we give them. If we place them in this cell into a solution that is 200 milliosmol, we can look at the effects that the solution has on the cell. So we can see right now that the solution is not as concentrated. It doesn't have as many particles just in the solution. We see that the cell has more. So what is going to happen is water is going to want to go into the cell. So we have water going into the cell to get these numbers to balance. So once these numbers balance, it will be at a resting state, but it does have effect on the cell. This is going to cause the cell to swell. So we can term this solution here, we can term it to be hypotonic. A hypotonic solution will cause the cell to swell, and if it's enough difference, it will cause the cell to lyse. If we place our cell into a 300 milliosmolar solution, there will be no water movement, and we can term this isotonic. All right, my last video cut out right towards the end. I was talking about tonicity. If the solution has a higher if it has a higher tonicity, or if it has a higher osmolarity of solutes that cannot permeate the membrane, then water is going to come out, and then this solution is going to be termed hyperosmotic. The cell is going to shrink. I wanted to talk about, just really quick, the effects of other molecules in a solution. If we had glucose in a solution, we know that the body uses glucose. Glucose is going to be taken into the cell very rapidly, and it's going to be metabolized. If we have a solution that has 500 milliosmoles of glucose, we can determine this to be a hyperosmotic solution compared to the cell. But a funny thing happens, like I said, when glucose goes in the cell, it gets metabolized, and this is going to happen to all 500 milliosmoles of glucose. It's going to go into the cell, and because it's metabolized, it doesn't change the osmolarity inside the cell, so that's going to stay at 300. But because it's all left the outside of the cell, this is going to be at 0. So we're going to have 0 osmoles of glucose left in the solution. Fluid is going to rush into the cell trying to balance this out, and this is going to be a hypotonic solution. All right, my last video cut out right towards the end. I was talking about tonicity. If the solution has a higher if it has a higher tonicity, or if it has a higher osmolarity of solutes that cannot permeate the membrane, then water is going to come out, and then this solution is going to be termed hyperosmotic. The cell is going to shrink. I wanted to talk about, just really quick, the effects of other molecules in a solution. If we had glucose in a solution, we know that the body uses glucose. Glucose is going to be taken into the cell very rapidly, and it's going to be metabolized. If we have a solution that has 500 milliosmoles of glucose, we can determine this to be a hyperosmotic solution compared to the cell. But a funny thing happens, like I said, when glucose goes into the cell, it gets metabolized, and this is going to happen to all 500 milliosmoles of glucose. It's going to go into the cell, and because it's metabolized, it doesn't change the osmolarity inside the cell, so that's going to stay at 300. But because it's all left the outside of the cell, this is going to be at 0. So we're going to have 0 osmoles of glucose left in the solution. Fluid is going to rush into the cell trying to balance this out, and this is going to be a hypotonic solution.